greatly acknowledge for inviting me to this very wonderful conference. So today I would like to talk about some research that I'm now going on in my groups. So especially there, as many professors, you and me talking, there are many target materials in the field of the industrial biotechnologies. But today, I would like to concentrate on the two materials, target materials. One is bioethanols, and another one is lactic acid. I would like to produce these kind of materials directly from the cellulosic biomass. So today, I would like to concentrate on that topic. So I'm from Kobe. Probably you don't know so much about the Kobe, but the Kobe is one of the center for the Japanese sake productions. So I heard that in Korea also you have a very strong industry in the alcohol brewing and amino acid productions. But in Japan also we have a very nice sake production industries nearby cities. So this is in sake brewing we use these two kind of microorganisms, aspirin saliva and bees. So this production is very different from the bioethanol productions, but nearby cities we have a very good accumulation of the industries. So today we would like to produce ethanol from cellulosic biomass. So probably you know that cellulosic biomass is consisting of these kind of the components. For instance, lignin 15 to 25%, and hemicellulose 23 to 32%, and cellulose 38 to 50%. So we would try to convert these materials to the bioethanols very efficiently. So if you think about these compounds, the cellulose is of course consisting of the glucose. So if you just hydrolyze cellulose into glucose, it's very easy to utilize for the microbial systems. But hemicellulose part is consisting of the many sugar components. For instance, xylose is a major component of this hemicellulose. So if you try to construct a very efficient bioethanol production system from this kind of cellulosic biomass, your microbial system should utilize all these sugar materials and convert these sugar materials into the ethanol. So that is not an easy task. And another very important factor is cost. So you should develop a very cost-effective conversion systems. So that is a very difficult point of these topics. So probably all of us know that there are so-called sugar platforms. So try to convert this biomass sugar part into the biofuels or building blocks. So we heard about this in this morning. So for this to achieve this process, first we should pretreat this biomass and saccharify this biomass into the mixed sugars, and then we should convert this mixed sugar to these biofuels or chemicals by the bioprocess. And then finally, in some, for instance, plastic or these kind of materials, we should further convert by the chemical or biochemical process. So to reduce the cost, it's very important to integrate this process because this process consists of many steps. So to reduce the cost, it's very important to construct some kind of integrated bioprocess. So that is a very important point. So I took these figures from the homepage of the Oak Ridge National Laboratories. They drew very nice pictures on these topics. So they call this is consolidated bioprocessing. This is a kind of integrated bioprocessing. I took this one from the homepage of these laboratories. And DOE constructing 3 biofuel centers in the United States. And this very nicely explained the systems. So in conventional systems, biomass native plants, they call, pre-treated. And then solid-liquid separation, because by the pre-treatment, hemicellulose part was solubilized and we can get the pentose sugars. And then pentose were fermented to the ethanols. And on the other hand, cellulosic part are still solid. So enzyme were produced, and enzyme added to these cellulosic materials, and these cellulose were hydrolyzed by this enzyme cellulase, and these hexose were fermented to the ethanols. Like these current systems consisting of many steps. So in the case of the consolidated bioprocessing, this is a modified plant, and reduced no pretreatment, and then biomass was directly converted to the ethanols by this consolidated bioprocessing microbes, CBP microbes. So of course I think this is a most cost-effective way. So just if you could successfully develop this kind of a very super microbe which can directly convert this biomass into the bioethanols, then of course this is the most cost minimum way. So of course in between there could be many different process, but probably future directions is this way. I agree these pictures. And we also have doing this research to these directions. So if you think about the CB consolidated bioprocessing, this is reported by Leading from the United States. There are two approaches for this construction, these CBP microbes. So he defined it that the native strategies. This is introduction of the ethanol fermenting pathways to the cellulose producing microbe, such as clostridium strain. So these strains are naturally producing the cellulose like cellulose. And if we can successfully introduce ethanol fermenting pathways, we can construct a very efficient CBP microbes. And another strategy is recombinant strategies. Production in this strategy, production of cellulose in ethanol fermenting microbes, like yeast or zymonas. So we are working in this under this concept. We are utilizing the yeast cells and try to produce the celluloses in these yeast cells. So as I explained later on, we are taking these approaches and what we call arming technologies. So we try to express and concentrate celluloses on the surface of the yeast strains. So this is quite similar. The concept is basically similar to this clostridium cellulose. They are producing the cellulose on the surface and accumulating cellulose on the surface. This is a kind of analogical strategies. So I would like to explain about our approaches like this. So this is the concept of arming technologies. So this is, for instance, any kind of microbial cells. So we can construct this kind of display technologies in any kind of the microbial cell systems. So in these systems, we try to display the cellulose target enzymes on the surface of the yeast cells. So like the analogy is like this. So like the Buddha cells and many arms. So this is what we call arming technologies. So if you can accumulate enzyme on the cell surfaces, so you can combine the power of the enzymes and the power of the microbial cells. So you can improve the productivities by metabolic engineering of the cells, and you can improve the cellulose hydrolyzing abilities of enzymes by the combinatorial bioengineering approaches. So like this, you can combine the power of the enzymes and the microbial cells. This is a kind of platform technologies. So we try to develop new types of these efficient cell surface display systems and try to apply that for the production of the bioethanols or lactic acid and so on, directly from the cellulosic biomass. So this is a couple of examples of the cell surface display systems in yeast. So just the concept of this kind of expressions concept. So here we have a promoters of the yeast systems, and here we have a secretion signal sequence here. And then here we have a genes encoding enzymes, for instance, cellulose to be displayed. And here we have a 3-half of the alpha-glutin genes. This is a well-known cell surface gene in protein in yeast. And at the C-terminal ends, here we have a GPI anchor attachment signal sequences. So if we have these anchor proteins, and if we fuse this anchor protein with the target enzymes, and if we express these fusion proteins, during transportation to the cell surface, this kind of structure of the GPI anchor attached to this signal sequence, and then this fusion enzymes were displayed on the covalently attached on the cell wall. So like this, just make this kind of fusion proteins together with the anchor protein, then we can efficiently display on the cell surfaces. And another approach is like this. So we try to use the kind of cell wall binding proteins. In this case, we are utilizing the, some protein for flow 1. These proteins were physically attached on the cell wall. So just utilize that kind of cell wall binding proteins and make a fusion expression system with these proteins and our target proteins. So like this is completely another approaches. So I would like to say that we can adopt many different types of approaches to accumulate the enzymes on the cell surface. So if you think about, for instance, yeast strains for the consolidated bioprocessing, so we accumulate many different types of the cellulose on the surface of the yeast cells, and then we put in the metabolic pathway of the pentose, for instance, xylose, and then we can establish this kind of the very efficient yeast strain which can directly utilize the cellulose and hemicellulose, or pentose, and efficiently produce ethanol from all these components from the cellulosic biomass. So the advantages of cell surface display system is very simple. If you can combine the enzymes on the cell surfaces, if you try to establish this kind of continuous fermentation systems, here we have a cell separator, and this is fermented. Just by the physical, this kind of segmentations, you can very efficiently separate cells together with enzymes. So you can recycle cells and enzymes simultaneously. So you can construct this kind of continuous fermentation system. And also, if you try to construct repeated fermentation systems, also we can separate very easily cells together with enzymes. So like this, it's very easy to construct this kind of fermentation system. And especially yeast, some yeast has a flocculation ability. So it's very simply, we can separate these by flocculation, sedimentations. And another advantage is like this. As I mentioned, we can adopt this combinatorial engineering approaches. So if you display the enzymes from this kind of the library, DNA libraries, and then if you find out some interesting enzymes, then you can make some combinatorial libraries. Then you can select your target cells which display your very interesting enzymes, and then you can select these cells by high-throughput screening method using the nanochan barriers or this kind of the flow cyte methods. And then you can directly apply that cells for the industrial productions because this is a cell. So cells, if this cell has enough performance, you can directly utilize these cells for the large-scale production. So in that sense, it's very straightforward method. And of course, we can apply this kind of cell surface displays technologies for many different applications. But I don't mention so much, but anyway, we can apply this for the many different purposes. So if you think about the cellulose hydrolysis, we need several enzymes. For instance, in this case, N-glucanase initially hydrolyze these amorphous regions, and CDH further hydrolyze, and these two enzymes simultaneously further hydrolyze to the cellulose saccharide, and then the glucosidases further hydrolyze this one to the glucose. So like this, at least we need a couple of enzymes for the hydrolysis of this cellulose. So to make a CPP microbes, we should display combine these enzymes on the cell surface. So as a first examples, we have tried to, because in these cases we should combine our systems with some pretreatment. As a first examples, we try to combine our systems with the concentrated acid hydrolysis pretreatment. The combination is very important. So in this first approaches, this in a concentrated acid hydrolysis, we utilize very high concentration of sulfonic acid. That is not environmentally friendly approaches, but this is very practical approaches. So we try to combine this one with our systems. So in these systems, the lignocell wood chips actually, it was hydrolyzed by the concentrated acid, sulfonic acid pretreatment, and then sulfonic acid was separated by chromatographic separations, and sugar mixtures was fed into the fermentation systems. Even though this is a very hard pretreatment, but still we have many oligosaccharides containing in this hydrolysate. So we display just beta glucosidase on the surface of these yeast cells, and for the xylose conversions, we express these xylose converting enzyme systems inside the cells. So we combine this xylose fermentation pathway together with this beta glucosidase display on the cell surface. So this is very simple, easy first examples. And the display system is very simple, something like this. So this is an intracellular expression system of the 3 enzymes, which is necessary for the conversion of xylose to the ethanol. And we display this beta glucosidase utilizing, for instance, in this case, alpha-glutamine display systems. So we co-transform this expression system into the yeast cells and try to apply this system for the conversion. The conformations of the cell surface display is very simple. We utilize the immunofluorescence microscope technologies. So if you can successfully display your target enzymes, beta glucosidase or whatever, then if you add the primary antibodies against this cell, of course this should bind. And then if you add a secondary antibody like this, the cells was fluorescence level. So just by observing this fluorescence microscope or FACS flow cytometer analysis, then you can confirm the successful display of your target enzymes like this. So then we confirm the fermentation abilities. This is just just fermentations of the just model xylose system, 50 grams per liter xylose, and because we express the conversion enzymes, and like this, the enzyme, yeast systems very directly, efficiently convert the xylose to the ethanols, and also. This is a cell bios, this is a model of oligosaccharide. This is a result of cell bios fermentations. And in this experiment, we culture the cells and put cell into this medium, and the fermentation is going on. And like this, cell bios is very efficiently assimilated, and we can very efficiently produce ethanols like this. So like this, if you display beta glucosidase, just beta glucosidase, you can assimilate this cell bios directly, this kind of oligosaccharide to the ethanol. If you take a best expression system, this fermentation speed is exactly the same as the glucose. So like this, we can very efficiently utilize this kind of the oligosaccharide directly. So this is a real example of the applying the application of our arming system to the lignocellulosic hydrolysate by the concentrating sulfonic acid. So like this, the most part of the hydrolysate is glucose and mannose, these kind of simple sugars. But it's also containing this kind of cellulosic saccharide. Even in that case, it's impossible to completely hydrolyze the cellulose into the simple sugars. So if you compare the fermentation by the wild type yeast and our arming yeast strains, then you can see that the very big increase of the yield of the ethanol. This is ethanol yield. So like this, if in the case of the this kind of very hard pretreatment case, we can improve the yield of the ethanol very much by just displaying the enzymes on the cell surface. So this is actually continuous fermentation. We did some pilot scale experiments. This is the picture of the continuous fermentation systems. Here we adopted the two fermenter systems. Here we have a first fermenter system. Here we have a cell separator and back to these fermenters, and this is second fermenters. So like this, we can directly ferment, or hydrolyzate of the wood chips. So if you think about the further systems, concentrated sulfuric acid hydrolysis system is not so good. We should simplify the pretreatment, and we should adopt some environmentally friendly pretreatment systems. So right now we are concentrating on the hot water pretreatment systems. So that is much more environmentally friendly. So we try to combine that kind of simple pretreatment with our arming technologies. So for that purposes, we should improve the cellulose and hemicellulose assimilating abilities of these arming yeast cells. So again, we need a couple of enzymes. We should express these many different types of enzymes simultaneously on the cell surface. So we try to express these three enzymes, these three AGCBH beta-glucosidase at the same time on the cell surface. This is just a simple experiment result. And here's this is a confirmation of cell surface display by the fluorescence labeling. So you try the different types of the antibodies against the BGL and the EG and the CBH. And then here we can see that by the old types of the antibodies, we can observe the fluorescence of the cell surface. That means that just we integrate three different types of the cell surface expression system of these enzymes, then we can confirm the co-display of these enzymes. So like this strategy is not so difficult. So then, if we can successfully co-display these enzymes, the cellulose was converted to glucose by the synergetic and the sequential reactions, and glucose was further converted to ethanols by the yeast cells. So this is a result of direct ethanol production from the amorphous cellulose. In this case, we just utilize amorphous cellulose as a sole carbon source, and we grow these yeast cells, and putting this yeast cell to this amorphous cellulose mediums. So this is the fermentation results, and this is the fermentation time, and the total sugar, this amorphous cellulose direct decrease like this, and we can directly produce ethanol from this amorphous cellulose as a sole carbon source. And the important thing is that yield of this ethanol is more than 90% of theoretical yield. So like that, this is a kind of model experiment, but we can directly and efficiently produce the ethanol directly from this kind of amorphous cellulose. So like this, by combining the very efficient and cell-based systems, we can directly convert solid cellulose to the ethanols. So another example is lactic acid productions by utilizing this kind of cell surface display systems. So in this case, we utilize the lactic acid bacteria. So of course we can utilize E. coli or many other microbial systems, but in this case we utilize lactic acid systems. So if you think about the lactic acid fermentations, starch, in this case, because cellulose is very difficult, so as a first example, we try to convert these starchy materials directly to the lactic acid. So if you think about the lactic acid production from starch materials, we should convert this hydrolyzed starch, and then the lactic acid bacteria should convert that glucose to the lactic acid by the lactic acid fermentations. So in the new approaches, we try to integrate these steps. Then to do that, we display the amylase, in this case, on the cell surface of the lactic acid bacteria. So I'd like to show you that because this display system is applicable for many different types of the microbial cell systems. Very simple platform technologies. So there are various types of the cell surface proteins which are available to construct display system in gram-positive bacteria like lactic acid bacteria and corynebacteriums like this. So, for instance, polypeptide glucan binding proteins like ACMA, these kind of proteins, or transmembrane protein like PGSA, or these many different types of the cell wall binding proteins are candidates for the construction of the cell surface display of the gram-positive bacterial system. So very simple, I would like to explain that. So, if you think about the lactic acid bacteria's cell surface display system, the concept is quite similar to the yeast display systems. So here we have a promoter systems. Here we have anchor systems like a PGSA, ACM. As I mentioned, ACM is a peptide glucan binding proteins, and this PGSA is a transmembrane proteins. This kind of, just to make this kind of anchor, use this kind of anchor proteins and make a fusion with the proteins to be displayed. Like this kind, just to make this kind of the expression system. So and transform this system to the this kind of gram-positive bacteria. So here I would like to show you the one examples. So amylase is the enzyme to be displayed, and this CA, so-called CA, is a peptidoglycan binding domain of the ACMA. So just to choose this kind of the domains, and we make this many different type of the tandem systems, because we expected that if you increase these CA domains, we can increase the binding abilities of the cell surface and make this kind of the expression systems. So as we expected, if we increase the cell wall binding domain, number of the cell wall binding domains, we can observe the very more strong binding on the cell surface. So like this, we can improve the binding abilities on the cell surface. So the important thing is that, so in this case, we can fuse the target protein, both C-terminus and N-terminus, because this is a kind of just a binding protein, physically binding on the cell surface, and then we can fuse our target protein to both sides. And if you look at this, in this case, enzymes has very high activities. But if you fuse the enzyme on the other side, then enzyme loses the activities. Like this, depending on the fusion side, we can, sometimes the enzyme loses enzymatic ability. So that in that sense, it's very important to have appropriate display systems. This is another example of utilizing a PGSA, a transmembrane type proteins. And in this case, also, we can very successfully display the alpha amylase on the cell surface. So like this, you can choose many different types of the cell wall binding proteins, and we can construct this kind of cell surface display systems. So this is actually fermentation results of direct lactic acid fermentation directly from the starch. The starch decreases like this, and lactic acid would produce directly. So like this, if you can construct this kind of the enzyme display microbial systems, you can directly ferment, for instance, in this case, lactic acid from the starchy materials. So of course, to further increase productivities, of course, you should utilize this kind of metabolome analysis and transcriptome analysis, as Senator Lee told us. So we should further improve the productivities. But it's very important to combine this display system together with this kind of metabolic engineering approaches. So finally, I would like to acknowledge all my students and arming systems and funding agency. Thank you very much for your attention.